In a previous video, I introduced us to the RasMol package and demonstrated that by double clicking on the RasWin file here, that opens the software. And I proceeded to look at the COVID spike protein just very briefly in that particular video. Now, I want to go in rather more detail now into looking at a second more straightforward protein. I am therefore once again going to open the RasMol window, and as previously we'll notice that in fact this black window with the pull down menus at the top is not the only way we have of importing commands. If we go here, we can see as well the RasMol command line. So as I did previously, I'm going to maneuver these around on the screen to maximize the amount of space that we've got available for the visualization window. And I'm going to import into this window a straightforward protein, ribonuclease, a very extensively studied protein. And as that protein lands into the window, it can be seen in this wireframe view. It is a very straightforward protein, as I hinted there, and one of the reasons it's been heavily studied is because it's so small. So if I put show sequence into the command window, we can see that in fact ribonuclease is only 124 amino acids in length. So it ends at valine 124. It starts at lysine 1. Now actually that's not necessarily the case. That if we look at a protein, we will see the whole of the sequence present. Sometimes you might see it starting with amino acid 4 or 20 or something of that sort. And that's usually an indication that there was a section at the end where the structure couldn't be resolved because it was too flexible. And if it's possible that that's actually been intentionally cleaved off so that you were able to form a crystal, if that was the way that the structure was generated. So we always begin with this wireframe view. It's not particularly informative unless you're got a canny eye for that kind of thing. So immediately I'm going to look at this from the display menu in the backbone mode. And that's clearly very much more straightforward. As the name implies, we are now looking just at the backbone of the protein. But things I want to look for here in particular, I can see the end, so I can click on the end here, just with the mouse. And in the command line window, we see that that's lysine 1. So I know that that's my end terminal end of the protein. If I maneuver it around, I can see the other end is here. And if I click on that, I can confirm that that is valine 124. So that is our C terminal end of the protein. So that's a more straightforward view than looking at the wireframe. But actually, it's not particularly informative for us. As we've mentioned previously, going to ribbons tells us very much more. And if we go for colour and colour that by structure, then we can see straight away that our ribonuclease has 3 short alpha helices, these pinky purple helices, and a couple of small sections of beta sheets. So one here made of 3 strands and one over here that looks like apparently it's made of 4. So we can start to see some more structural information quite clearly there. I'm going to go back to the backbone view for the next step of this video, and I'm going to choose from the color menu from the command line. I can actually choose colors there as well. There are a number of accepted colors for the system, and for the time being I'm going to put color cyan in there. So that's a color I don't take. So let's change it all to being this light blue color. Now what I can do then is to start to pick out different characteristics of amino acids and look for those in particular. So I can now put select hydrophobic, and that chooses 575 atoms. If I go to the I can then type in color pink, and we can see where those are within this sequence. If I put in if I put in select hydrophilic, which you might think was the opposite command, you'll notice that in fact it says syntax error. So although RasMol is quite straightforward, it is quite picky about the things that you say. And the opposite of select hydrophobic is not select hydrophilic. It is in fact select not hydrophobic. I need to check my spelling in a second there. Again, that's something it doesn't like if you spell things. Select not hydrophobic. Okay, so I seem to have got that correct. And I can now choose colour blue from my menu. And we start to see some information there, but this is of limited value to us. What's going to be more useful if I now go for select protein? Now in fact, I'll go for select all for the time being. Now, the reason for doing that is because any command that I type into the command line now will only follow what went previously. So if I've done select not hydrophobic, then anything that is hydrophobic won't be picked up by these next commands unless I've gone back to pick everything up, essentially. So I put select all in again. And now if I go to the menus at the top here, if I go to display space fill, we start to see that in fact the pink bits that we saw, the pink sections, are not particularly obvious on the surface of the protein. There's a dominance of the blue colour, so of hydrophilic positions. Now that's obviously what we'd expect. Hydrophobic things tend to be buried away inside a protein, and so that is as we would expect. Actually, the picture that we're seeing here is a little more biased towards the hydrophilic than we might imagine, because it's actually captured some water molecules as well. So to make those less visible, I'm going to choose select water in the command line, and I'm going to change those to display in wireframe. And so we see that there are now fewer of those visible. We do start to get a better feel now for the three-dimensional shape of this particular protein. And what we can see is that there is a very definite groove running through the middle of the molecule here. And we can see that that's got some hydrophobic groups lining along that particular position. And we'll come back and look at that more in a subsequent video. For the moment, what I want to do is just to pick out a different characteristic about ribonuclease. So I'm going to select all again. And I'm now going to put everything back into backbone view. And for simplicity, I'm going to colour that magenta. So it's removing any of the characteristics that I've picked out so far. So this is just our backbone. And what I'm going to do now is to pick out the cysteines in particular. So one of the reasons that ribonuclease has been quite widely studied is because it has within it 8 cysteine residues, and that they form 4 disulfide bonds within the molecule. And we can get a feel for where those are. So I'm going to put in select cis, so that's going to pick out just our cysteines, and then I'm going to colour those yellow. And we can see where they start to show up within the structure here. And already, even in this backbone view, we can see that we have an obvious pair here, a second pair here, a third over here, and a fourth down here. So we can see where the pairs are occurring. But there are ways of looking at this which are more informative. Remember, I've still got just the cysteine selected at the moment. So if I now go to the display menu and pick sticks, then we can see where those pairings are occurring. And so they become more obvious. However, we can add further information if we now go to the colours menu and we choose from there to go CPK, where what we're now showing is the colour of the sorts of elements that are present there. So if I was to zoom in on one of these, in the middle here, we can see that we now have a red clear, which is oxygen. We've got a blue for the nitrogen. We have a yellow for the sulfur, and we've got the carbons in grey. And so we start to get more structural information. And what we can see now rather than the general colour yellow that I chose, yellow now clearly indicates sulfurs, and we can see the positions of the 4 different disulfide bonds that occur within this molecule. So that's a very straightforward introduction. I do encourage you to dabble with the RasMol package, to look at the display menu, to look at what the different effects of the different options there are, and similarly with the colour menu, and possibly to look at some of the others as well. But for the time being, I'd focus just on display and on colour. Now, I will come back and look at ribonuclease again in a second video, which will be looking more at the mechanism and what we can learn about the mechanism of the enzyme by studying it in this way.